Hello, everyone. My name is Patrick Hu. I'm head of cancer medicine at MD Anderson. I'd like to welcome everyone. Thanks so much for coming to this extraordinary day. It's a real privilege for me to introduce Jim Allison, my good friend and colleague, who I've known for many years, also a fellow musician, as this year's winner of the Nobel Prize in Physiology or Medicine. As a melanoma and tumor immunologist myself, I've really seen firsthand the impact that Jim's research has had on patients. I've had many patients now in my clinic who are alive and well, having had advanced melanoma because of the way that Jim took basic science findings to the clinic and has helped many people. Now these research is now being applied not only to melanoma, but to many other cancers as well. It's really revolutionized our cancer therapies today. So really congratulations to Jim. Yeah. Jim has been at MD Anderson now since 2012. He really came back to MD Anderson. He's from Texas. He grew up in Texas. He grew up in Alice, Texas. He used to tell me that as a kid, he would come from Alice to Houston in the summers to cool off. So we're really happy to have him at MD Anderson, where he is continuing to do cutting-edge research and applying now his findings to many other kinds of cancers, understanding immune resistance and how to improve these therapies yet further. Today is such a momentous occasion for Jim and his family. Pam Sharma, who also herself is an outstanding clinical investigator, immunotherapist, and son Robert Allison, who's here. So congratulations to the whole family. On behalf of the faculty and staff, our president, Peter Pisters, from MD Anderson, we congratulate you on this much-deserved honor. We're very proud, but also very happy for all of the patients that you've helped, all of the patients that you've helped now, and the patients in the future that will further be helped by your tremendous research. So again, congratulations, Dr. Allison.
Thanks, Patrick, for those kind words. I want to start off by saying I'm still in sort of a state of shock, and this is all sinking in. So I'll try to make some sense while I'm talking here. But it's really wonderful this morning to wake up, and my son called me at 5:30 and was the first to let me know that I'd won this prize, and I want to thank him for that, and Pam Sharma, my partner in life and in science. We work together now trying to improve immunotherapies. Also want to thank a long line of students and postdoctoral fellows and colleagues at MD Anderson and at UC Berkeley and at Memorial Sloan Kettering over the years that have helped bring this work to where it is, and also the patients who, especially the early trials, who put themselves at some risk when taking a drug which could potentially unleash the immune system to eliminate cancer, but also possibly to do harm. And luckily those effects are not all that serious, or at least can be managed. And so it's really turned out to help quite a number of patients. And I guess the reason I'm really thrilled about this is I'm a basic scientist. I did not get into these studies to try to cure cancer. I got into them because I wanted to know how T cells worked. And once we showed that this molecule CTLA-4 was the first cell-intrinsic negative regulators of immune responses that immediately occurred to me and some of the people in my lab that maybe we can use this to unleash the immune system to attack cancer cells. And so I'm lucky enough, as a basic scientist, to see my work actually end up 20 years later, actually really helping patients. And I think that I want to say this modestly, but I think that this award, I was told by the Nobel Committee when I was called this morning that this was the first prize they'd ever given for cancer therapy. And so they've given some for cancer, the causing cancer before, but this is the first time for cancer therapy. And I'd like to just give a shout out to all the patients out there who are suffering from cancer, to let them know that we are making progress now. We can get durable responses in a fraction of patients with many different kinds of cancer. And of course, we still have a lot of work to do. There's some kinds of cancer. Well, first of all, it's a fraction of patients who respond. In melanoma, it's about 60%. We need to get that up as close to 100% as we can. With other cancers, it's somewhat lower. We need to get all of those, the fraction of patients that respond, and also bring these therapies to some kinds of cancer that have not yet responded, including glioblastoma and pancreatic cancer, the other things that you hear about in the news all the time. And we're trying to do that at MD Anderson in the immunotherapy platform there that Pam Shorman and I run together, where we're trying to understand mechanisms of what goes on exactly in patients, both when it works and when it fails, and understand the reasons for both so that we can do rational combinations in the future to bring the benefit to more patients. So we've still got a lot of hard work to do, but the optimism comes from the fact I think we know the basic rules now. We've just got to work hard and learn the details and develop more personalized treatments for the majority of patients. And like I said, the good news is I think there's optimism to think that we can do that, but it's going to take a while. So I guess I'll stop there and take any questions that you might have. Who's okay? Right.
For cancer someday. I wonder if you could just describe your mindset at the time, and what was it that got you up every morning and going into the lab when it was just purely for understanding some basic thing in biology?
Well, the dirty little secret, I think, of most scientists is that what really motivates us at the end of the day mostly is the desire to know something that nobody else knows, and hopefully something that's important, to be the first people to really figure out something complex. I mean, ever since I was a kid, I like to solve problems, and I think that's what keeps scientists going is, you set yourself towards a task. I mean, when I started working on T cells, nobody knew how they worked at all. Didn't know the structure of the molecule that recognized the things that they would attack, didn't know the molecule, didn't realize that actually that there were additional molecules that were required to both, accelerator pedal and brakes, and then other modulators down the road. So it was really that kept me going most of the time, because science, is a long and frustrating road to get in. There's no instant gratification of it. It's just the whole nature of science is trying to prove your hypotheses wrong. The better you do that, the better you're doing. And so you've got to be, comfortable with a lot of failures to get there. But I've had, a lot of cancer in my family, and my mother passed away from lymphoma when I was 10 years old, and she'd had radiation therapy, and one of her brothers died of lung cancer and had chemotherapy, and I saw the ravages of those kinds of treatment. And as I got into immunology, I always had the hope, and read a lot of papers, particularly from Lloyd Old, who I was lucky enough to have as a mentor, who in the early 60s began working on using the immune system in animal models. So it was always in the back of my mind. It wasn't the reason I was doing the work, but whenever I could, I would take a rest, from thinking about the problem and look at our data and say, what does this tell me that I could use to treat cancer? And that was one of those moments when we figured out that CTLA-4 was the brakes on the immune system. I said, well, let's just disable the brakes and see if that will allow the immune system to attack cancer. And it did. And so we did the animal experiments, and several years later did the early clinical trials. And we have patients now, actually one patient from the phase 1 trial in 2001, they got a single dose of anti-CTLA-4. A patient with metastatic melanoma is still around almost 19 years later. So these are very durable responses. And again, they're in a fraction of patients, but that's what we got to work on now. We know we can do it. We just got to learn how to do it better. Rather long-winded answer. Sorry about that.
Any other questions?
Yes, I'm with John Friends Press AFP. What was your reaction when you got the phone call? Tell us where you were.
Well, I was in the hotel that the Cancer Research Institute, which I've been associated with for many years, was having a major meeting up in Times Square with it's an international meeting with people from immunological societies all over the world, and Europe and the United States anyway. But so I was at that meeting. Pam Sharma won an award there, gave a talk yesterday, and so I was in the room, and my son called at 5:30, and then pretty soon I started getting calls from a lot of other people, and then a bunch of people came to our room, to the door, and beating on the door at 6 in the morning with champagne, and so we had a little party early this morning. It was really great with my friends, all of whom work in this field in one way or another, at the meeting. So it was tremendous.
Well, what was your emotional reaction to winning the Nobel Prize? I mean, when
Yeah.
did you speak to the committee? Did they call you?
For some reason they had trouble and didn't reach me until after I knew about it from my son and my friends. But they finally got me, and we talked about it. And that was when the committee member, said we've never given one for cancer therapy before. I guess it still, as I said, it still hasn't completely dawned on me, except that I'm a basic scientist, and to have my work really impact people is just one of the best things I can think about. I think it's everybody's dream, and I've been lucky enough to, do work that is benefiting people now, and I'm still trying to, figure that out. It's quite a shock to learn this at 5:30 in the morning. So, but it's as I said, I think it's every scientist's dream to have, first of all, do fundamental work that's important, but then to have it translate into helping people is just all that much more better. And have it recognized by the Nobel Committee is, I think, going to bring attention to the issues of the importance of basic science, and that there is hope for cancer patients.
Hi, my name is Miho. I'm from Japanese broadcaster called Nippon TV. Congratulations on receiving this amazing award. And I just wanted to ask you actually about Professor Honjo, who also received that award today. Could you tell us, if you've met him, if you worked with him?
Yes, I know Tasuku Honjo very well. We've worked together. Well, not really worked together, but known each other and discussed science, and not on this, but on other topics since the early 80s, actually. And so he made this discovery of this molecule PD-1, which was the second checkpoint discovered. And then about the time that, we had a problem understanding why CTLA-4 only worked in about 20% or so of patients. And so there are a number of possible explanations for that, but of course the most interesting one was that it's not the only checkpoint, that there's another one, and Honjo, his work brought PD-1 to the fore, and Now we know those two together are extremely powerful. CTLA-4 has a response rate in melanoma by itself of about 22%, but if you add PD-1 blockade to it, it goes up to about 60%. And so it just helped, show that this approach was good, and to broaden the impact and broaden the possibilities for helping patients.
Could you actually, sorry, one more question. So could you give us any comments or message to him if you can?
Sorry, the last
Could
one?
you give us any message or comment to Professor Honjo?
Yeah, just give him my best and say I look forward to seeing him in Stockholm in December. We shared the Tang Prize several years ago, so we go back a little bit on this too.
Yes. Malcolm Ritter from the Associated Press. Just to go back to your story of how you heard, it's not clear to me. You said there was your phone rang at 5:30, but you didn't answer it, or there were other calls. I'm a little confused.
No. The phone rang at 5:30, and it was my son calling me.
Oh.
And spoke just for a few seconds because there was a call from Sweden, and I thought, oh well, because he'd already told me that that I'd won, but it was a reporter from Sweden, so.
Oh.
And I talked with him briefly, and then my friends all showed up, and we started celebrating. And it was a while later. I still don't understand why the reporter had my number, but the committee didn't. But anyway, they finally found it and called me. I don't know what time it was, 7 or something like that.
So you heard the news from your son?
Yeah.
Okay. Thank you very much.
Kieran Weiser, Fuji TV. You seem like a very modest person and not really wanting to take a lot of own personal merit in this award or own personal boasting. As a scientist, how does it feel to be able to share this award with a fellow scientist, let alone a friend like Dr. Honjo, who you respect very much?
It feels very nice. As I said, when we did it, the CTLA-4 work, it was kind of just, I don't want to say one-off. That's not quite what I mean, but it was a singular thing, and one of the worries was that it was going to be stuck at that 20-something%. And I'd known Honjo from the early days when we were both trying to figure out the T-cell antigen receptor structure, which we had done, and we considered working together to clone the genes for a while, but that's when I first met him. But he's done a lot of work on antibodies, a lot of areas of immunology he's worked on, and made many really important contributions to fundamental immunology, particularly in B cells, but more recently in T cells as well. So it was great to have him come along and identify another one of these molecules, as I said, that can broaden the applicability of the general theme of checkpoint blockade to cancer therapy.
One more question, if that's okay. What do you think makes you and Dr. Honjo such great friends as both colleagues, but also away from the workplace?
Away from, sorry?
Things that are unrelated to work? Do you have any special personal memories?
Yeah, a little bit. I mean, not all that much because he's in Japan and I'm here, but we've met particularly at scientific meetings. We've never golfed together. He plays golf. I don't play golf, so so that's never happened. But we've had a cordial relationship for, like I said, 30 years or so.
Sure. Thank you.
Do we have any more questions in the room? Otherwise, we'll take some from the phone. Oh, yeah.
Yes.
Oh, yeah, Bob Langworth with Bloomberg News. Congratulations. I just wanted to ask, why do you think if scientists have been working on immunotherapy, particularly the vaccine area, for many, many years before your work, why were companies and cancer doctors not focused on this checkpoint blockade area until you did your work? Just kind of explain your kind of perspective and why that was less researched.
Well, I think the idea of using the immune system to treat cancer goes way, way back. The problem was it started being applied before we really knew how it worked. And so, for example, with cancer vaccines, I think that looking back on it in light of what we discovered with CTLA-4 is that, after the first vaccination, since the CTLA-4 is turned on by the antigen receptor stimulation, you vaccinate you're really increasing the signal through CTLA-4. So every time you do it, you're turning the immune system off. You're beginning to, dampen the signals down, and people just didn't realize that. And I think that another thing is we just don't know how to we still don't know the proper way to vaccinate, to get therapeutic responses. Obviously, we could do a great job against microbes, but in terms of really instantly mobilizing the immune system to go after something like cancer, we still have a lot to learn there. But basically, I think it was a lot of the early immunotherapies were they just started with insufficient knowledge to know the details of the circuitry that we were working with.
One last question here from in the back. Just in the simplest way you could explain it in the shortest time period, what for average people, what does your and your colleagues' discovery mean for the future of people who may be suffering from cancer?
I think that what we've learned, again, to put it very simple, is that there are built into the immune system these inhibitory circuits that stop the immune system at a certain point so that it doesn't hurt normal tissues. And but that also keeps it from attacking tumor cells as effectively as the T cells might. And so we found a way to temporarily suspend the brakes and let them take off. So after many years of resistance, I think the world, the cancer field has begun to accept immunotherapy now as the fourth pillar, along with radiation, surgery, and chemotherapy, of cancer therapy. What we have to look forward to is that, unlike the other three, immunotherapy can be used in combination with the other three. And I think that what we're looking forward to is combinations in the future, not just of multiple checkpoints, but of checkpoints with radiation, checkpoints with chemotherapy, checkpoints with genetically targeted small molecule drugs. To really, I think that immunotherapy is going to be part. It's not going to replace all those others, but it's going to be part of the therapy that essentially all cancer patients are going to be receiving in 5 years or so. And they're going to be curative. They're going to be curative in a lot of patients.
Move to instructions for the folks on the telephone.
At this time, if you would like to ask a question, please press the star and 1 on your touch-tone telephone. You may withdraw your question at any time by pressing the pound key. Once again, to ask a question, please press star and 1 on your touch-tone telephone. And we will pause for a moment to allow questions to queue.
Have some water.
We do appreciate your patience, and we will take our first question momentarily. And we will go ahead and take our first question from Ivan Perone with ASP. Your line is now open.
Dr. Allison, can you talk about the other pillars precisely that you mentioned? How important do these conventional therapies remain? And what should patients think and tell the doctors about? Should all the funding go to these areas, to immunotherapy as opposed to all the therapies?
Yeah, well, first, as I said, I think that there is still a place for the for the more conventional therapies. There are some cancers that we don't have, so far immunotherapy hasn't worked in. As I mentioned, glioblastoma and pancreatic cancer, those are big challenges. But the whole process of getting the immune system going after cancer starts with some tumor cell death. And that alerts anyway, without making it complicated, that causes inflammation. That starts a whole cascade of immunological events. And so if you could kill cancers with radiation, or you can kill cancer cells with chemo, what we know now is that the traditional way of giving high doses of chemo or high doses of radiation and trying to kill that last cancer cell, every last cancer cell, is no longer the goal and should not be the goal of those other therapies. You can moderate their use and just try to kill enough kill enough tumor cells to initiate the immune cascade and then come in to finish the tumors off by activating, sustaining T cells. And so, but I don't think all the funding by any means ought to go into that. I mean, we know that with the small molecules, for example, those drugs have been developed with in vitro assays and then in xenografts, tested in xenografts of human tumors growing in mice that don't have immune system. So in most cases, we have no idea whatsoever what those drugs do to the immune system. But we need to know that if we're going to begin to combine them with immune therapies. The same for chemotherapy. We need to know, many chemotherapies can probably work. We need to know their effects on the immune system, though, and if they are immunosuppressive, to come up with staging, dose scheduling that allow them to be used together. But I would say that what we need to do is to continue to fund basic research, because that's where these ideas came from, not from trying to kill cancer, but from trying to understand how the immune system works. And if everything's translational and targeted on a given area, pretty soon there's going to be nothing to translate because there won't be any new ideas. So the big jumps come from the basic sciences, and I think we got to keep that in mind always. We can't just say everybody's got to be trying to cure a disease. It does not work that way.
For James, I apologize. I just arrived. You probably already said what I'm going to ask you, if you don't mind. My name is Fabio from Brazilian Global TV. Just would like to ask you a few words about how you're feeling by getting this prize, and also what kind of change did your findings contribute to the cancer treatment and perspective to the patients all around the world?
Yeah, well, I guess as far as I'm feeling, I'm still kind of in a state of shock, actually, from getting up this morning and learning of this. But the thing that gratifies me is I do consider myself, well, anyway, I used to be, maybe not anymore, but a basic scientist who was just trying to learn about the immune system and happened to come across something that we could exploit due to the help of a lot of really, really bright students and fellows and everything. I think what this means to cancer patients is, if I dare use the word cure, which I know is a very dangerous thing to do, but we have patients now that are 18 years out since they get one round of therapy and they're 18 years out, 14 years out, whatever. And I think those patients can be considered cured. And I think that that's the hope that these kind of therapies work, because the immune system, after all, differs from conventional therapies in several ways. One is that once you've got T cells, you've got them for the rest of your life, and so they don't go away. If you give a chemotherapy drug, it's gone in hours. And so you really have to keep giving it until you kill every last tumor cell. You don't have to do that with the immune system. The cells will be around and can keep coming after the tumor and changing as the tumor changes to recognize new antigens on the tumors. As the problem with cancer is the tumors are so heterogeneous because of the genomic instability that they're very hard to treat. But that makes them, just easy targets for the immune system, because the immune system loves heterogeneity, and the more antigens, the better. The more mutations, the better. And I think that, again, what this offers, we've still got a long way to go. We're curing patients now. The idea is to really, really learn in detail how it works and bring these benefits to more patients. And it's a challenging thing, but what we know now that we didn't know 10 years ago is that it can be done, at least in some patients. So we've got to, we got a grasp on it, and we just need to, keep working.
Do you already know how many patients already benefited from this therapy?
Not really, but I think hundreds of thousands have been treated. So it's tens of thousands, I'm sure. I don't know. I've been trying to ask Bristol-Myers Squibb, who, helped us get anti-CTLA-4 on the market, and nobody seems to know, but it's a big number. 4 years ago, for example, there were 5,000 patients at least who they had 10 years follow-up on, so the real number is somewhere way above 5,000. just with ipilimumab. And then with PD-1 and the new combinations and stuff, it used to be that this therapy was given to late-stage patients who had no other options. Now it's the first choice for melanoma, for example. A patient starts on immunotherapy rather than chemo or even the genomically targeted drugs. The only patients who get those are the patients who fail immunotherapy now, and I think that's the way many cancers are headed.
Okay, we have one more question from the phone.
So we will take our next question from Tina Say with Science News. Your line is now open.
Hi, I was wondering if you can tell us a little bit more about why certain cancers seem to respond to these checkpoint inhibitors better than others do?
Yeah, there are several reasons. One is there seems to be an association with the total mutational load, the mutational burden. Those that have a lot of mutations, we know now with the immune system, it's sort of the T cells in the case of cancer are preoccupied with targeting proteins that are made by the mutations that occur during the cancer process itself, not just the drivers, as they're called, that cancer biologists have been looking for, but has the potential to recognize all the passengers as well. So melanoma, for example, can have 3,000 or so mutations per cell. So that's 3,000 potential targets to see. But you get down, it ranges actually, it's a range from a few hundred to thousands. But if you get down to something like prostate cancer, there may be 30 expressed mutations. Now that doesn't mean that absolutely is unresponsive. We recently found in a study that we can see T cells that recognize mutations out of even a small number of mutations. But that's part of it. I mean, the more mutations, clearly, the more likely you are to get one or more that the immune system can recognize well. Another one is that a lot of tumors are what we call cold, or they're immunological deserts. There's no T cells in them, and so until you get the T cells in, you can't really do anything. We know now also, though, that CTLA-4 will drive T cells in tumors, but then it also induces expression of more checkpoint inhibitors, such as the ligand for PD-1. And so then you have to start thinking about giving combinations to deal with it. There's some new ones coming along. So, and then also in some tumors, it looks like they have active methods of suppressing, multiple methods of suppressing the immune system and protecting themselves from cancer. And so there's no simple answer, but some of these things we can overcome. We know how to overcome now. Other ones we don't know now, and it's going to take a lot more research to understand how to do that.
Okay, thanks. We have time for one more question.
Thank you.
Anyone? Okay. I'd like to thank you all for coming today. I appreciate it. Any final remarks?
Okay. Thank you all.